Thank you very much, Peter. Those very generous remarks. And thank you, Jonas and Jan, for inviting me and organizing all of my activities yesterday afternoon, today, and tomorrow. This is my second visit to Gothenburg. I was here 2 years ago for a more ceremonial presentation. It was cold and wintry, and this time it's warm and beautiful. And today it's full of science, and that's the best part of today, is hearing what everyone else in the field is doing. So this afternoon, what I'm going to talk about is work that's gone on in my lab now for the last few years. I'm a new entrant into the field of extracellular vesicles. Most of my career, I've studied intracellular vesicles, but much of the approach that we took in trying to understand the mechanism of protein sorting intracellularly, we've employed in trying to understand how RNA is sorted into vesicles that are secreted from cells. And I'll tell you about that work, which was initiated by a brilliant graduate student, Matt Shurtleff, when he joined my lab just 6 years ago. So let's begin. This is a slide that you've seen versions of, and I needn't repeat very much about it, other than to point out, as is obvious to all of you and in the discussions that we've already had this afternoon, there is a diversity of vesicles that are secreted by cells. They are exported by a variety of pathways, and we probably don't even fully appreciate the complexity of the many different species that are being produced, which is why when Matt joined my lab 6 years ago, we felt that it would be necessary to try to purify a vesicle by sequential purification steps in order to understand how molecules like small RNAs could be acquired by a particular vesicle species, which is not to say that it will be a unique mechanism. I suspect there'll be many other paths for the production of vesicles that contain different RNA species. So when Matt joined the group, we had already been working on mammalian cells. Most of my career I'd worked on yeast cells, but we were working on mammalian cells, and just for convenience, we started with HEC293, a cell line that many of you know about that we used. And we designed a procedure, a three-step procedure for the isolation of CD63 containing vesicles, which is highlighted on this slide. We start, as all of you do, by sedimenting first at low speed to remove debris, and then at high speed to enrich small vesicles. But that material, I was certain, was fairly crude, and so we needed to do additional steps. The next step was to take that material, resuspend it in 60% sucrose, put it at the bottom of a sucrose step gradient, and then sediment to equilibrium by flotation, where we found that CD63 equilibrated at a position between 20 and 40% sucrose. Membranes were also fractionated elsewhere on that gradient, but CD63 was enriched at that interface. And we didn't think that that material was pure at that point, so we then used a polyclonal antibody against CD63 immobilized on beads. We absorbed the interface membrane fraction to those beads, and then sedimented at low speed and washed with buffer. And we began to characterize that material for its protein content, but mainly for its small RNA content. And we focused initially on microRNAs. Matt did a deep-seek microRNA analysis of the 600 or so different species that are produced by HEC293 cells. And here is a Venn diagram of the distribution of those RNAs. Among the majority of the microRNAs are found both in cells and in exosomes, not particularly enriched, but distributed in both membranes. About 10% of the microRNAs are enriched in the immobilized CD63-containing vesicles. But what was particularly striking about these vesicles was that only a very small number, perhaps a handful, are very highly enriched. They are the outliers shown here, including one called MIR144. But another less abundant microRNA called MIR223, we estimate to be 1,000-fold enriched in these CD63-containing vesicles. Really quite striking. So when you think about it, the cell has gone to considerable trouble to sort that RNA, to distinguish that RNA in a small number of additional RNAs to be packaged into these EVs. Now, it could be that the cell is just desperately trying to get rid of those microRNAs, to dispose of them. That's certainly a formal possibility, but it seems more likely to me that the cell is deliberately selecting those RNAs to be transferred to another cell. Now, I don't have anything to add to that discussion. It's a very important question, one that we're addressing now in the lab. But I think it'll be really very important to try to define a role for microRNAs in extracellular vesicles. And I, for one, am not particularly satisfied with any of the demonstrations that claim that microRNAs can be transferred in EVs. Certainly, microRNAs can move from one cell to the next, but whether that's mediated by EVs, I think, is still an open question, one that we certainly wish to address. Now, another really surprising thing about the handful of microRNAs that are sorted in this way is that in comparing the sequences of these 4 or 5 different species, there was no obvious primary sequence similarity at all. So how is it that a small number of microRNAs are recognized and sorted when there's no obvious similarity? Either there's some structural element that's shared among these RNAs, although people who study the structure of small RNAs tell me they are not folded by themselves, or there are multiple signals, and we suspect it's the latter. There are probably multiple RNA sorting signals, and I'll tell you about some experiments that have defined one that's responsible, we believe, for the sorting of miR-223. So now, to show that these very highly enriched RNAs really do track during the purification procedure, we evaluate the relative level of these RNAs in the membranes as they are subjected to this sequential purification procedure. And you can see that there is a progressive enrichment in the sorting of miR-223 and of miR-144 at each step in the purification. So there really is some efficient purification going on in this fractionation scheme. We could show that these RNAs are inside of the vesicle. We use a very simple principle, that is to demonstrate that the RNAs are resistant to exogenous ribonuclease unless the membranes are suspended in the presence of a non-ionic detergent. That's shown here. Both of these microRNAs really are protected. They are sequestered in the interior of the membrane, and they are readily degraded by exogenous ribonuclease, which will become important in a moment as I tell you how we then have tried to approach the question of the mechanism of RNA sorting. Now, in much of my career, and really largely from the training I received in Arthur Kornberg's lab as a graduate student, I've felt that in order to understand a molecular mechanism of a complex biological process, it's essential to try to recapitulate that process in vitro. And in vitro, I mean not in tissue culture cells, I mean in broken cell preparations, where you isolate membranes and separate them from cytosolic proteins and then mix them back together in an effort to reconstitute in the test tube some essential process. And so Matt was up to that challenge, and we devised a very simple idea to detect the sorting of miR-223 into vesicles that form when membranes from HEC293 cells are mixed with cytosol from the same cells in the presence of ATP. And that is shown in the diagram at the top. We break cells open gently by a mechanical trituration process, squeezing cells through a narrow gauge needle, and mixed membranes, presumably some of which are endosomes in the preparation, with, in this case, a chemically synthetic form of mature miR-223, and add to that cytosol from the broken cells and ATP. Membrane transformation events often require hydrolyzable ATP. We then incubate the mixture, in this case at 30 degrees for 20 minutes, after which the membranes are centrifuged at low speed, exposed to exogenous ribonuclease to degrade any RNA that has not been taken up and sequestered within a membrane. And then the membranes are broken with detergent and a ribonuclease inhibitor, and then the RNA that's been sequestered is measured, amplified, and quantified by qT-PCR. And in the initial experiments, Matt was able to show that he could see about 8 or 9% of the exogenous miR-223 protected in a vesicle that was sensitive detergent and where the ribonuclease was protected unless detergent was present. And he could show that sequestration required both the presence of membranes and cytosol. If either were omitted from the incubation, no such protection was observed. Further, he showed that the reaction was stimulated about twofold by the addition of hydrolyzable ATP. So it and required incubation at a roughly physiologic temperature. If the sample simply held on ice, nothing happened or very little happened. So that gave us some encouragement. And one signal, for instance, for miR-223 is shown here. In this case, about 9% efficient protection of the microRNA compared to a parallel incubation held on ice. But the most important criterion, which is shown here, is that this process is RNA selective. If you do the same experiment with a purely cytoplasmic microRNA, in this case we chose one called miR-190, it's not secreted from cells, it's just inside the cells. If you do the same incubation, very little of the exogenous miR-190 is taken up in exactly the same incubation. So this really gave us confidence that we were measuring something of physiologic meaning. We were sorting RNAs into vesicles formed in vitro. Now, one obvious means by which an RNA could be distinguished from another one is by the intervention of some kind of RNA binding protein. And so Matt devised a very simple scheme to allow us to fish out from the cell-free reaction any RNA binding proteins that adhered to miR-223 and which became enclosed within a vesicle. And that's shown here. It's the same incubation that we've done previously, that I showed you in the previous slide. But in this case, Matt included a chemically synthetic miR-223, but with a 3' biotin group, providing a hook that would allow the RNA then to be removed after the incubation by binding to streptavidin beads to examine any RNA binding proteins that came along for the ride. So the incubation was done as before. Membranes were sedimented, washed, treated with exogenous ribonuclease. The membranes were dissolved, and in this case, then the samples were mixed with streptavidin beads, proteins that adhere, the RNA that adheres to the beads, brought along RNA binding proteins. The beads were then washed. Proteins that bound were removed by high salt and subjected to mass spec. And there were several RNA binding proteins that he found, but one stood out at the top of the list, a protein called YBOX1, or YBX1, a protein that had been seen previously in extracellular vesicles. Indeed, a publication demonstrated, I think I have that in my next slide, a publication demonstrated that the YBOX1 protein is secreted from cells, even though normally it has some intracellular function. It's been associated with messenger RNA splicing. But some fraction of the YBX1 protein is secreted, and importantly, the secreted form is sequestered within a membrane because it's resistant to a protease unless non-ionic detergent is added. So that's a simple criterion to establish YBX1 as at least partly secreted in a vesicle. We obtained a polyclonal monospecific antibody against the YBX1 protein, and we could then examine its distribution during our fractionation of the CD63 vesicles. And further, we could examine its incorporation into a vesicle during the course of the cell-free incubation. And these results are shown on the next slide. So here is the analysis of the CD63 immobilized fraction. There is CD63. There is the YBX1 protein, and several other known exosomal membrane proteins are present in this fraction. Interestingly, although flotilin has been described as an exosomal membrane protein, and simply it did fractionate along with CD63 on the step density gradient, Matt noted that flotilin is not associated with CD63, at least by this criterion, because it flowed through the immobilized antibody beads. So this may represent a distinct exosomal species, one that would bear its own purification and characterization. Now, in the next experiment, Matt repeated the very same experiment that I've described to you, where he adds exogenous in this case biotinylated miR-223, and then he examined the requirements for the formation, for the detection of a membrane in which the YBX1 protein could be detected by immunoblot. That's shown on the right. It's very simple incubation, just as before, with membranes and cytosol powered by biotinylated miR-223, incubated at 30 degrees for 20 minutes, after which the sample's sedimented, the membranes are washed, they're exposed to ribonuclease. Ribonuclease inhibitor is added, and then detergent is added to dissolve the membranes. The sample is then mixed with streptavidin beads to bring down all the biotinylated miR-223, and then the beads are treated with SDS sample buffer, run on a gel, and immunoblotted. And there it is, YBX1, as expected, comes along for the ride. Importantly, it's not seen when an incubation is conducted without cytosol, nor is it seen if an incubation is conducted without membranes, or if the incubation is simply held on ice during the 20-minute period. And of course, if the biotinylated miR-223 is not added, little, if any, of the YBX1 protein is incorporated into vesicles. Now, just as an aside, and I'll come back to this in a moment, what was surprising about the mass spec analysis was not so much that the YBX1 protein was there, but that a major microRNA binding protein, Argonaute, was not there. And this was of some concern to us, and even more concern to the referees of our paper, who were skeptical that we had failed to find the Argonaute protein. But we went back, and I'll show you one piece of evidence later on, and we were able to show that although Argonaute can be detected sedimenting along with membranes from conditioned medium of HEC293 cells, it is simply not in vesicles. It can be readily separated from vesicles on a buoyant density gradient. So, and unfortunately, there have been claims in the literature that Argonaute is associated with exosomes, but unfortunately, it's all based on co-sedimentation in an initial 100K spin. So that is one of many reasons I'm very concerned about using that alone to characterize things that are in exosomes. Okay, so YBX1 comes along for the ride. It's bound to miR-223, but is it important in the sorting event? And for that, we made a CRISPR homozygous null of HEC293 cells. YBX1 is an important protein for the viability of an animal, and a double knockout is an early embryonic lethal event. But fortunately, it can be, you can create a homozygous null HEC293 cell line, grows, as far as we can tell, perfectly normally. And we could show that in the homozygous null, little, if any, detectable YBX1 protein. I'll mention in a moment there are two paralogs of YBX1, YBX2 and 3. They are relatively immunologically distinct, but there may be some partial functional overlap between these paralogs. Nonetheless, we could do two kinds of experiments with this cell line. I'll only show you one. We could evaluate whether the YBX1 null cell blocks the production of exosomes. It does not. And we can evaluate whether the exosomes that are produced by the null cell are depleted of miR-223. It does to some extent. It's a partial depletion, and I'll show you that in a moment. Equally importantly, we were able to show that when you take YBX1 null cytoplasm, not YBX1 null cytosol, that cytosol is quite deficient in packaging miR-223 in our cell-free reaction. So that's demonstrating biochemically that that protein is not only coming along for the ride, but it's actually required for the sorting of miR-223 in our cell-free system. Here's the in vivo experiment. We looked at YBX1 null cells. We examined not only the EVs that are produced by the null cells, but we also looked at the accumulation of miR-223 and of miR-144 in cells that lack the YBX1 protein. That data is shown above the line. There's about a 3- to 4-fold increase in the accumulation of both miR-223 and of miR-144 in these cells. miR-144 is also depleted from EVs, purified from the culture medium, again about 4-fold. Surprisingly, there was only about a 2-fold diminution in the secretion of miR-223 in EVs produced by these cells. Matt showed that that's because the second copy of YBX1, the paralog YBX2, seems partially to serve a redundant function. And he demonstrated that by taking the YBX1 null cells and ablating the expression of YBX2 by siRNA. And under those conditions, both miR-144 and miR-223 are reduced about fourfold in their secretion in EVs that are produced by these cells. Now, in collaboration with Alan Lambowitz at the University of Texas, we've done a more thorough analysis of all of the nucleic acids associated with purified vesicles. And it was quite interesting that the most abundant species in the purified vesicles are not microRNAs, but full-length tRNAs, yRNAs, and vault RNAs. They are quite substantial contributions to the chemical concentration of RNA. And interestingly, each of these three major species appeared dependent on a YBX1 for their incorporation into vesicles. They are reduced about fourfold in the RNA found within vesicles isolated from the YBX1 null cells, which is not to say that all RNAs are depleted. We found some small messenger RNAs in the purified extracellular vesicles. And these small intact messenger RNAs seem not to be dependent on YBX1 for their incorporation into vesicles in the YBX null cells. Now, hidden in this data is a really surprising fact, and that is when we evaluate the concentration of tRNA comparing wild-type cells to YBX1 null cells, there's a substantial difference in the amount of tRNA. And that's shown in these two bars. So this is a level of tRNA in intact wild-type cells. This is the same measurement in YBX1 null cells. There's about a twofold difference between these two. And one interpretation of that observation, by no means the only interpretation, but one interpretation is that during the 36 hours that we cultured the cells to harvest EVs from the conditioned medium, those cells that were deficient in YBX1 may have accumulated so much more tRNA than they ordinarily would. There may be, during that 36 hours, as much as half of the tRNA content of the cell being secreted in EVs. So that really was quite a revelation, to me at least, that cells would be so massively secreting RNA molecules like tRNAs in extracellular vesicles. Now, we did a survey of lots of other nucleic acids using the criterion for those that are highlighted in green of RNA species that are resistant to exogenous ribonuclease unless the vesicles are suspended in triton. So all of these species in green are individual species that are clearly enclosed within the intact EVs. We found DNA associated with these purified EVs. They are DNA molecules that have protein coding sequences. They code for link RNA. They have some antisense coding sequence. But at least for these EVs, isolated from HEC293 cells, all of that DNA was accessible to exogenous DNase in the absence of detergent. And we believe thus that that means that the DNA is simply adventitiously bound, absorbed to the surface of exosomes. It may have no functional significance, although that will really be hard to establish. Now, as this work developed and as we published this work in the past 2 years, a new graduate student, Moraima Timosh Diaz, from Peru, joined my lab and followed on Matt's work to evaluate species of EVs that are produced by a different cell line, a breast cancer cell line. And she found that she could resolve two different species based on a buoyant density separation on a linear optoprep gradient, and that's shown here. There was, she found a population of vesicles that sedimented approximately to the same buoyant density as the vesicles that Matt isolated from HEC293 cells. We refer to these as high buoyant density membranes, and they are characterized further by coincident localization of endosomal related membrane proteins that fractionate at this position on the density gradient. There's also a distinct lower buoyant density population that has a slightly different spectrum of known exosomal membrane proteins with some overlap, but with a clearly distinct peak, clearly two separable populations. She took these two separate populations and did a microRNA survey analysis to see if there were species unique to each individual buoyant density fraction, and that's shown here. We could see in the low buoyant density membranes some enrichment of several microRNAs, although not a very striking enrichment, maybe 10 to 20-fold enrichment. And yet, in the high buoyant density species, the similar high buoyant density vesicle that Matt had isolated, but now from a different cell line, yet another spectrum of a small number of microRNAs that were very highly enriched, thousands of fold enriched. One of them that I'll tell you about in a moment is miR-122, a microRNA that we did not find in EVs from HEC293 cells. Now I want to return to a slide that I skipped over that emphasizes a point that I made a moment ago, and I want to emphasize again. If you evaluate the distribution of membranes and RNAs and RNA binding proteins across this gradient, you can show quite convincingly that two of the proteins that are necessary to mature microRNAs, dicer, which cleaves the loop from the stem loop precursor, and then argonaute that adheres to mature microRNAs, are both found at the bottom of the gradient. They represent ribonucleoprotein particles that are not buoyant at all. They sediment to the bottom of the tube. So, I mean, it's kind of interesting that argonaute and dicer are seemingly secreted by these cells. They may have some other mechanism, but they're not secreted in vesicles. They're readily separated from vesicles. Okay. Now, so given this result, this now new microRNA that's, again, completely different sequence than miR-223, Moraima decided to have a look using the procedures that Matt had developed to see if she could recapitulate the sorting of chemically synthetic mature miR-122 into vesicles in a cell-free reaction. And she was able to do that, but she had a very surprising result right from the outset, where she demonstrated that miR-122, though it's packaged efficiently, seems not to be dependent on the YBX1 protein for its packaging. And that data are shown here. First, she was able to repeat Matt's experiment, showing that miR-223 is efficiently incorporated in vitro, and yet it is completely dependent on YBX1 in the cell-free reaction. It's always very comforting when two different graduate students repeat the same experiment. I was delighted with this result, but even more delighted with the result on the right, where she demonstrated that miR-122 is packaged, perhaps not quite as efficiently, but still clearly above background, and yet its packaging seemed to be much less dependent on YBX1. So clearly, different RNA binding proteins may be invoked in the sorting of different microRNAs into vesicles in the cell-free reaction, and as you'll see, also in intact cells. Well, this then leads to the next obvious experiment, which was to repeat Matt's experiment, but using a biotinylated derivative of chemically synthetic miR-122, which she did, using his exact same protocol. She did the cell-free reaction with biotinylated miR-122 and cytosol and membranes and ATP. She harvested the membranes, treated with ribonuclease, dissolved the membranes bound to streptavidin beads, etc. And she found 3 different RNA binding proteins represented in the fraction associated with miR-122. They are nucleolin, the Lupus LA antigen, and nucleophosmin 1. She then went through each of these systematically to develop a depletion strategy. It turns out that it wasn't possible to make knockouts of any of these genes, at least not in this breast cancer cell line. So she used a CRISPR-I technique that allowed her to deplete one at a time each of these 3 proteins in cells that were grown over the course of several days under depletion conditions. And she found that one of them, called Lupus Law, seems to be required for the sorting of miR-122 in vitro and in vivo. And let me show you the data for that. Here first is the depletion of the Lupus Law antigen. It was first detected as an autoantigen in lupus patients. It's an RNA binding protein. It has some obscure functions in cells. It's certainly essential for the viability of cells. It was possible to deplete it quite substantially, in contrast to a control protein, and possible then to take cytosol from the depleted cells and compare it to wild-type cytosol in a miR-122 packaging reaction, the data of which are shown here. So in a concentration curve experiment where one varies the concentration of cytosol, it was clear that wild-type cytosol responds linearly over the course of this range of protein concentrations, whereas the Lupus Law depleted cytosol was much less active in packaging miR-122. And so she examined, at this point, in an incubation with 3 milligrams per mil of cytosol, either wild-type or mutant, and confirmed that the lupus depleted cytosol is grossly deficient in the packaging of miR-122, whereas in the controls where samples are held on ice, the formation of protected RNA is simply not observed. She then evaluated Lupus Law depleted cells, where they were depleted over the course of several days. She harvested conditioned medium, isolated a crude EV fraction, and evaluated its content of miR-122 and compared it to the content of miR-122 in cells from which the Law antigen had been depleted. And these results are shown here. In the EVs isolated from cultured cells, there is a little over a 2-fold depletion of both miR-122 and another one of the microRNAs that was enriched in EVs from this breast cancer cell line. And there is a corresponding 2-fold increase in the intracellular concentration of these microRNAs. They accumulate because, to this extent, because they're not being secreted. So for 2 different, entirely different RNAs, miR-223, we have an RNA binding protein that's required, YBX1 protein. And for miR-122, we have a seemingly different requirement, in this case for the Law protein. Now, very recently, in sequencing the entire pool of microRNAs isolated from EVs produced by the Law depleted cells, she's found a diminution in the secretion of miR-223 as well as of miR-122. So the requirements for these RNA binding proteins are complex and not yet clear, but they seem to cooperate to some extent in selecting RNAs for sorting into vesicles formed at least in the cell-free reaction. Now, in the final minutes, I'll return to the question of what the sorting signals may be that allow these RNAs to be so efficiently separated into vesicles, at least in the cell-free reaction. I told you there's no common sequence element between these RNAs, so it was hard for us to think how we might go about addressing the question of a sorting sequence. But a young colleague of ours, Nicholas Ingoglia, suggested that we could do an experiment, a variation on a now classic experiment in RNA evolution, where you create a giant random pool of RNA oligomers, and you sort among this pool in vitro for RNAs that have a particular structure or function. It's called CELEX. And so Matt adapted the idea of CELEX to a large random pool of RNA oligomers to look for sequences that could be packaged into vesicles formed in his cell-free reaction. The logic is shown here. It's very simple. We take a huge pool of RNA oligomers and mix it with membranes and cytosol and ATP, conduct the incubation as you've now seen a half a dozen times. At each point, the RNAs that are not incorporated are removed because they're degraded by ribonuclease. The membranes that protect the incorporated RNAs are then dissolved. In this case, they are converted back to DNA templates, which are transcribed, and the RNA pool, now refined, is returned to a fresh sample of cytosol and membranes, and then recycled over and over again 4 times to try to enrich for those sequences that can be protected by this sorting event. David Melville, a postdoc in the lab, developed an algorithm that allowed us to scan octameric sequences that are enriched during the course of this CELEX series of incubations. The pool was clearly refined during the course of this selection. Still, at the end, it was a very complex set of sequences. But among the sequences that emerged from this CELEX, a particular sequence was particularly interesting. A run of C residues found in 2 independent replicates of 4 cycles of sorting. In one instance, a run of 3 C residues, another instance, a run of 4 C residues, with a very high level of confidence in each replicate pool. And it then did not escape our attention that there are 4 C residues near the 3' end of mature miR-223, and there are no C residues at all in the RNA that we chose as our control, miR-190. So this obvious difference suggested one additional experiment, which was to test whether those run of C residues was both necessary and sufficient for sorting RNA in the cell-free reaction. So the last experiment I'll show was that Matt created 2 derivatives, miR-223 lacking C residues, and a derivative of miR-190 into which he introduced 3 C residues near the 3' end. These 4 molecules were then subjected to a sorting reaction in vitro. And miR-223, in this case now very efficiently incorporated, but when the C residues are removed, it's no longer packaged in our reaction at all. miR-190, which is poorly packaged, as you saw some minutes ago, now becomes very efficiently incorporated when it is provisioned with 3 C residues. So this experiment then tells us, at least for the biochemical reaction, that the run of C residues are both necessary and sufficient for sorting. A new postdoctoral fellow in the lab, Liang Ma, has taken this experiment into intact HEC293 cells, and he's found that when he expresses a transgene form of miR-223 lacking C residues, it is no longer efficiently packaged into EVs by living cells. So we think this is a signal, at least for miR-223. But now, it's not found, this run of C residues is not found in the other microRNAs that are packaged. And in fact, surprisingly, miR-144, which is of anything even more dependent on YBX1 for its sorting in cells, has no C residues at all. So that means that there's another signal, likely another signal which Moraima is now looking at in miR-122. And I think the experiments are now obvious for how we will go about defining the various sorting signals and deciphering their RNA binding protein requirements. Ultimately, I'm most excited that we can reproduce this whole pathway in a cell-free reaction, because it means that we have the ability to fractionate the system, to ask which other proteins are required for this biogenesis event. Clearly, there'll be many other proteins that are responsible for budding a vesicle into an endosome or budding it from the plasma membrane. And what excites me most is our ability to use traditional biochemical means to try to answer these questions. So let me then summarize what we've learned, and even more importantly, what we haven't learned. We believe that ribonucleoprotein particles form in vitro. How miR-223 or miR-122 evades capture by the Argonaute protein that would allow it to be presented to a messenger RNA for control of protein synthesis. How that happens, we don't know. Maybe Argonaute is displaced by YBX1, or maybe some fraction, some small fraction of microRNAs simply escape being captured in the Argonaute complex. That remains to be seen. We want to know how the microRNA is recognized. We've found one sorting signal. Actually, Maraima's found another one now in miR-122. We don't understand how these RNA binding proteins relate to one another. One idea is that maybe there's some kind of complex of RNA binding proteins that form in association with the major RNAs that will be packaged. Some kind of a cluster of RNAs and RNA binding proteins may form in the cytoplasm. Perhaps even an RNA granule may form in the cytoplasm that can be enveloped by a membrane. That remains to be seen. Another open question that we have access to is how are the RNPs recognized by the machinery on the endosome that's responsible for sorting into the invagination that will give rise to an intraluminal vesicle. The machinery that's responsible for this reaction is well known in all eukaryotic cells. It was discovered by Scott Emmer studying this process in yeast. It's a series of proteins that are called the escort complexes. They act in sequence to recognize ubiquitolated membrane proteins that are sorted in the plane of the endosomal membrane and are delivered into the interior of a membrane invagination. We don't know if the escort machinery is necessary for the production of exosomes. The literature is actually somewhat contradictory on this point, but again, we have this cell-free reaction that we can use to discover that. Interestingly, there's a paper in the literature that demonstrates that the YBX1 protein is ubiquitolated, and there's a suggestion in this paper that its ubiquitolation is required for its secretion. And so it may be that YBX1 becomes ubiquitolated on the surface of the endosome and then is recognized by the escort machinery and gathered in along with other cargo molecules that may then be delivered into the interior in these vesicles. We want to know what the primary membrane source is for this budding reaction. I've drawn it as though it's the endosome, but topologically, what we've reproduced in vitro could just as well be budding of membranes on fragments of the plasma membrane. They are exactly the same topology, but obviously different membranes. And so one simple experiment that I'd love to do, not myself, of course, would be to have someone fractionate the membranes in our cell-free system to separate endosomes and plasma membrane and see which of these are more effective templates for the sorting reaction that we've developed. Well, here's a snapshot of the group from several years ago. I'll highlight the two most important people whose work I've described. This is Matt Shurtleff, a just spectacular graduate student who naturally then left. Broke my heart, but he left. He took his PhD, and he's moved now to Jonathan Weissman's group at UCSF, where he's a postdoc. But fortunately, there are many other able people he left behind, one of whom, Moraima Timosh-Diaz, is still active in the lab and has a year or two to go. Now, since I see the time is not up and I have a few minutes, I want to tell you about something that has consumed even more of my time and should be of great interest to you, and that is the question of how you and we and everyone decides where to publish your most important work. This is a subject of some controversy. I think there has been a really toxic influence in biomedical science that's developed over the last 15 years, and that is how our scholarship is measured at institutions around the world. The postdocs in the room will know that they favor journals that have a so-called high impact factor, journal impact factor, a number that is measured to, amazingly, 3 significant figures. A number that I will tell you is a phony number. There are many aspects of that number that do not measure scholarship. It was never intended to be a measure of scholarship. It was created many years ago to be used by librarians to decide which journals to subscribe to for their libraries. And the number even existed when I was a graduate student, but no one paid any attention to it. It's a measure, as you know, of the number of citations in a journal over a period of 2 years. It's a narrow snapshot of the influence of a journal. Now, some journals, particularly commercial journals, journals that are published by Elsevier and Springer Nature, use this number to promote their journals. And they have ways, they're very sophisticated, they're big businesses, they branded themselves effectively. They know how to manipulate the number to achieve a higher number, to give them seemingly greater impact. And I think, and I've argued in many forums over the last several years, that this has led to a toxic influence in biomedical science. And here's something that's not mine, it came from a review article. Cutthroat academia leads to natural selection of bad science. Nature, Science, and I will point to Cell. They publish very important papers, no question about it. But the number of papers in some of these journals and in their clones that turn out not to be reproducible is really quite striking. You may know that the biotech company Amgen, some years ago, conducted an internal study looking at what they call the most influential papers in cancer biology in the preceding years, papers that they might have relied on to develop cancer chemotherapeutic drugs, and they found that 85% of the key experiments in these papers could not be replicated. And that was a shocking result. I'm not sure how accurate it was. Unfortunately, Amgen didn't actually publish their findings. They didn't find it to their competitive advantage to do so. Nonetheless, this has been a serious concern in the biomedical community, but in particular in government circles, because they think they worry if they're spending billions of dollars to support our research, they'd like to at least know that it's reproducible. Well, I want to argue that some of this, I think perhaps a lot of it, is because of this ridiculous pressure to publish in a few small venues where the decisions are made largely by professional editors who are in the business of selling magazines. And they are, to a large extent, influenced by this phony number, the journal impact factor. Now, I'm often asked, as I travel around, if I dismiss impact factor as a measure of scholarship, how are we to decide what's going to be important work and who are we to listen to? What kind of evaluation scheme as an alternative to impact factor can be employed? And I was delighted to see last year a novel, really quite novel mechanism for evaluating scholarship that was published in Trends in Cell Biology. It's called The Cell Biologist's Guide to Fine Dining. It's like the Michelin Guide of journals. So in this model, an expert, in this case the maitre d' at a restaurant, has received a sample of food from an aspiring chef, and they've evaluated the quality of this food just as Nature would be evaluating the quality of your paper. And the maitre d' says, "Thank you for your contribution. We have now looked at your food and recommend that you take it elsewhere," just like what you get from a letter from Nature or Cell. Well, now, having used the criteria in this Michelin Guide to important cell biology, I was obviously eager to see how they evaluated certain journals without an impact factor. And I was particularly delighted that one of the entries was a description of eLife, the journal that I happened to edit. It's called, according to this Michelin-style review, "Super trendy new place with an organic menu, pride themselves on their ethical policy, backed from on high by some very successful restaurateurs, and beginning to be a victim of its own success. Prices are rising fast, and tables are becoming harder to come by." How about the competition? Nature and PNAS were evaluated together with Science, and the conclusion of this guide was, of these three journals, old world sophistication with menus that haven't changed in decades. The proprietors refuse to change anything, complacent in their past glory, and content in the knowledge that people will keep coming. It's almost impossible to get a table. The portions are tiny, and people don't generally enjoy the experience, but they do brag about it afterwards. PNAS which unfortunately I used to edit, has a backdoor for members-only dining, although it's considered a bit of a naff to use. And finally, of course, what about Cell? How do they fare in this new review system? Well, not so well, as I'll show you. Sleek and sophisticated upmarket Italian place that's openly run by the mafia, AKA Elsevier. The quality of the food is very good on the whole, and the menu is kept up with the times, but it's a bit of a guilty pleasure because you know you're lining the pockets of seriously dubious owners. Franchise affiliates, the clones, trade on name and carry the same caveats. So bear this in mind next time you choose your restaurant and where you choose to publish. Thank you.